If you are over 40 and your belly fat keeps growing no matter what you do, I need you to stop right now. Not because I have a dramatic announcement, but because what I'm about to walk you through could genuinely be the most useful 45 minutes you have ever spent trying to understand your own body. Because here's the truth that nobody in mainstream health media seems to want to say clearly. The majority of people over 40 who are struggling with weight gain, fatigue, brain fog, and stubborn belly fat are not failing because of laziness. They are not failing because they lack discipline. They are failing because they are working with an incomplete picture of what is actually happening inside their body. And when you have an incomplete picture, even your best efforts end up aimed at the wrong target. I have spent over 20 years working with patients dealing with metabolic dysfunction, insulin resistance, prediabetes, and chronic fatigue. And in that time, the single most consistent thing I have observed is this. The people who finally turn things around are not the ones who tried harder. They are the ones who finally understood the mechanism. Once you understand what is actually happening at the cellular level, the confusion clears, the frustration lifts, and the path forward becomes logical instead of overwhelming. So that is exactly what we are going to do today. I am Dr. Megan Foster, and in the next 45 minutes, I am going to take you through the real biology of what happens to your metabolism after 40, why the standard advice keeps failing so many people, what the research is actually showing us about the nutrients and signals your cells depend on, and what you can practically do starting today to begin shifting your body back in the right direction. There is a published study showing that people with the highest intake of one specific mineral had a 71% lower risk of worsening insulin resistance. Not 7%, 71%. And I promise you, by the end of this video, you will understand not just what that mineral is, but exactly why that number is so large, because the mechanism tells the whole story. Let us start at the very foundation. Because if we do not build this from the ground up, the rest will not land the way it needs to. Your body is an energy management system. Everything it does, every heartbeat, every thought, every movement, every repair process requires energy. And the primary currency of that energy system is a molecule called glucose. Glucose comes from the food you eat, particularly carbohydrates, but also from other sources the body converts. It travels through your bloodstream after a meal, and it needs to get into your cells to be used. Now, here's the critical part. Glucose cannot just walk into your cells on its own. It needs a signal. That signal is a hormone produced by your pancreas called insulin. Insulin travels through the blood and finds specific receptor sites on the surface of your cells. When it attaches to those receptors, a sequence of internal reactions takes place that opens the door and allows glucose to enter. When this works properly, it is an elegant system. You eat, glucose rises, insulin responds, cells absorb the glucose, blood sugar returns to baseline, and you have energy. The whole process is smooth, efficient, and largely invisible. You never have to think about it. But here's what starts happening after 40 in millions of people. The doors get stiff, not broken, not sealed shut, just progressively resistant. The insulin arrives and attaches, but the internal machinery that responds to that signal starts operating below its optimal capacity. The doors open slower. Less glucose gets in. Blood sugar stays elevated a little longer than it should. The pancreas notices this and responds by producing more insulin. More insulin is sent. More pressure is applied. The doors eventually open, but the system is working much harder than it should to achieve the same result. This is insulin resistance, and it is not a disease. It is a process, a slow, quiet, compounding process that can build for a decade before anything shows up on a standard lab test. And this is the part that I think is most important to understand. Because most people think insulin resistance is discovered when their blood sugar is high, but blood sugar is often the last thing to change. In the early and middle stages of insulin resistance, Your pancreas is overproducing insulin to compensate. Blood sugar stays in range because your body is working overtime to keep it there, but the underlying dysfunction is already creating damage. You feel it before you see it on paper. You feel it as fatigue after meals, as brain fog in the early afternoon, as cravings for sugar even when you have already eaten, as sleep that does not restore you the way it used to, as belly fat that accumulates specifically around the midsection and lower back, as a feeling that your body is fundamentally different now than it was 10 or 15 years ago. Those sensations are real. They are biological, and they are your body sending you an early warning signal that the system needs support. Think back for a moment to how your body felt after a meal when you were in your late 20s or early 30s. You probably ate, felt satisfied, had reasonable energy, and continued with your day. You could recover quickly from a heavy weekend. Losing a few pounds was inconvenient but manageable. Sleep generally recharged you. Now fast forward to today. A heavy lunch makes you want to lie down. Your afternoon energy crashes reliably around 2 or 3 in the afternoon. You wake up already thinking about coffee. The weight that used to come off with a few weeks of effort now barely moves after months. And the cravings, especially at night, feel like they have a mind of their own. That gap between then and now is not just aging. It is the accumulated effect of a system that has been under increasing stress without the right support. And the reason I'm spending time on this emotional reality is that most people blame themselves for it. They tell themselves they are less disciplined than they used to be, that they need to try harder, that this is what getting old feels like, and they just need to accept it. I want to be direct with you. That story is not accurate, and accepting it as truth keeps people stuck in a cycle that can actually be interrupted. Let me now walk you through the science of why this happens specifically after 40. Because there are biological reasons, not just lifestyle reasons, for why metabolism shifts the way it does. Understanding these reasons is empowering because it shifts the question from what is wrong with me to what is happening in my biology and how do I respond to it. After age 40, several things change simultaneously in the body that collectively put pressure on the insulin signaling system. Muscle mass begins declining at a rate of roughly 1 to 2% per year if nothing is done to preserve it. This process is called sarcopenia, and it is one of the most underappreciated drivers of metabolic change. Why does muscle matter so much metabolically? Because muscle tissue is your body's primary site for glucose disposal. When you eat a meal and glucose enters your blood, your muscles are the main destination. They absorb glucose, either burning it for immediate energy or storing it as glycogen for later use. The more muscle you have, the more efficiently this process works. When muscle mass declines, you lose capacity. The same amount of glucose now has fewer places to go. Blood sugar stays elevated longer. More insulin is needed to manage it. And the system comes under strain. At the same time, mitochondrial function changes. Mitochondria are the energy-producing structures inside every cell. They are sometimes called the powerhouse of the cell, but that description understates how complex they are. Mitochondria do not just produce energy. They are central to regulating how the cell responds to insulin, how inflammation is managed, and how well the cell repairs itself. After 40, mitochondrial efficiency tends to decline. Cells produce energy less effectively. And when cells are energetically stressed, their ability to respond to insulin signals is further compromised. Hormone shifts play a role as well. Estrogen and testosterone both have direct effects on insulin sensitivity. Estrogen, in particular, plays a protective role in how cells handle glucose. As estrogen declines in women approaching perimenopause and menopause, this protection weakens. Testosterone in men has similarly protective metabolic effects, and declining testosterone levels in men over 40 contribute to both muscle loss and reduced insulin sensitivity. These hormonal shifts do not cause insulin resistance on their own, but they reduce the buffer. They make the system more vulnerable to the other compounding factors. And cortisol, the stress hormone, becomes more chronically elevated in many people after 40, not because life becomes more stressful, though it often does, but because the body's cortisol regulation system, called the HPA axis, becomes less precise over time. Cortisol directly interferes with insulin signaling. When cortisol is high, the body is essentially being told to keep blood sugar available for emergency use. Insulin's job of clearing that blood sugar becomes harder. Fat storage, particularly in the abdominal region, is actively promoted. So, you have declining muscle, declining mitochondrial function, shifting hormones, and dysregulated cortisol. All of these are happening in the background, often without any obvious single trigger. And when they are all operating at the same time, the cumulative effect on insulin sensitivity is significant. This is why the standard advice of eat less and move more often fails people over 40. It treats a complex biological shift as if it were simply a math problem. Calories in versus calories out ignores the fact that the cells processing those calories are operating in a fundamentally changed environment. The hormonal context, the inflammatory state, the mitochondrial efficiency, the muscle mass, all of these determine what your body actually does with the food you eat, not just how much you eat. Now, let us go deeper into the specific factors that drive this process, because understanding them individually gives you specific places to intervene. The first major factor is muscle loss and metabolic capacity. We touched on this briefly, but I want to expand on it because there is an important dimension most people miss. Muscle tissue is not just a glucose storage unit. It is a signaling organ. When muscles contract during exercise, they release molecules called myokines. These are signaling proteins that travel through the blood and have effects on distant tissues. Some myokines improve insulin sensitivity directly. Some reduce inflammation. Some support the function of the pancreatic cells that produce insulin. Some even have effects on brain health. This means exercise, and specifically muscle contracting exercise, does more than burn calories. It actively sends corrective signals into the metabolic environment. It is communication. Your muscles talking to your liver, your fat tissue, your immune system, your brain, your pancreas. And the less muscle you have and the less you use it, the quieter that communication becomes. Research published in the journal Diabetes Care found that even a single session of resistance exercise improved insulin sensitivity for up to 24 hours afterward. Not because of calories burned, because of the hormonal and metabolic signaling cascade that muscle contraction triggers. This is why resistance training is not optional for people over 40 who are dealing with metabolic dysfunction. It is not extra credit. It is primary medicine. 2 to 3 sessions per week of exercises that genuinely challenge the muscles, whether that is weights, resistance bands, body weight, or any combination, creates a measurable and lasting improvement in how your cells respond to insulin. The second major factor is sleep and circadian biology. Sleep deprivation and insulin resistance have a closer relationship than most people realize, and the mechanism goes much deeper than just feeling tired. Your body runs on a roughly 24-hour internal clock called the circadian rhythm. This clock governs almost every physiological process in your body, including insulin sensitivity, cortisol release, hunger hormones, fat storage patterns, and how efficiently your cells repair themselves overnight. When this clock is disrupted, whether through insufficient sleep, inconsistent sleep schedules, or light exposure at the wrong times, the consequences are systemic. One study published in the Annals of Internal Medicine put healthy volunteers on a controlled sleep restriction protocol and found that after just 4 nights of sleeping, 4.5 hours, insulin sensitivity dropped by 16%. Another study found that sleeping less than 6 hours per night over time was associated with a 28% higher risk of developing diabetes compared to people sleeping 7 to 8 hours. But here is the part that goes beyond the numbers. During deep sleep, your body performs a kind of metabolic reset. Cortisol reaches its lowest point. Growth hormone is released, and growth hormone actively promotes insulin sensitivity and muscle repair. The gut performs maintenance on its bacterial communities. The brain clears metabolic waste products through a system called the glymphatic system that only operates properly during deep sleep. And inflammatory signals that accumulated during the day are resolved. When you consistently cut this process short, none of these things complete properly. You wake up with slightly elevated cortisol, slightly elevated blood sugar, slightly impaired insulin sensitivity, slightly higher inflammation, and slightly stronger hunger signals, particularly for sugar and refined carbohydrates. Because your body is looking for the quick energy hit it did not get from restorative sleep. Over months and years, those small daily deficits compound into significant metabolic damage. And there is one more layer to this. Poor sleep and insulin resistance form a bidirectional relationship. Insulin resistance worsens sleep quality, and poor sleep worsens insulin resistance. Once you are caught in this cycle, it can feel impossible to break from the outside. Addressing the biological factors that support sleep quality, including nutrient deficiencies we will discuss shortly, is often the first real crack in that cycle. The third major factor is the inflammatory environment. Inflammation has become such an overused word in wellness culture that many people have stopped paying attention to it. So I want to be precise about what we mean here and why it matters so directly for insulin resistance. Your immune system produces signaling molecules when it detects a threat. Some of these molecules, called cytokines, are part of the normal healthy immune response. But when inflammation becomes chronic and low-grade, meaning the immune system is partially activated all the time without ever fully resolving, these cytokines begin interfering with normal cellular function. Well, specifically two inflammatory cytokines called TNF-alpha and interleukin-6 have been shown in extensive research to directly block insulin receptor signaling. They essentially jam the internal machinery that processes insulin's message. The door receives the knock, but the lock mechanism has been chemically altered and responds poorly. This is not a theory. This has been demonstrated in cell cultures, in animal models, and in human clinical studies. Chronic inflammation is a direct mechanical cause of insulin resistance at the cellular level. Now, here's what makes this particularly challenging. Visceral fat, the deep abdominal fat that accumulates around your organs rather than under your skin, is itself an active producer of these inflammatory cytokines. It is not just stored energy, it behaves like an endocrine tissue, constantly releasing signals. And those signals include the very inflammatory molecules that block insulin function. This creates one of the more frustrating biological loops in metabolic health. Insulin resistance promotes fat storage, particularly visceral fat. Visceral fat produces inflammatory signals. Inflammatory signals worsen insulin resistance. More insulin resistance drives more fat storage. The cycle feeds on itself. Breaking this cycle requires reducing the inflammatory environment, not just restricting food. And the strategies that reduce inflammation most effectively include consistent physical activity, adequate sleep, a diet high in anti-inflammatory foods and low in refined and processed ones, stress management, and ensuring adequate intake of the nutrients that regulate immune function, including a specific mineral we will get to very shortly. The fourth major factor is what I call metabolic confusion from poor dietary timing and composition. Let me be clear first about what this is not. This is not about eliminating carbohydrates. Carbohydrates are not the enemy of metabolic health. The human body has used carbohydrates as a primary fuel source for our entire evolutionary history. The problem is not carbohydrates. The problem is the context in which we eat them and the form in which they arrive in our modern food supply. When you eat refined carbohydrates, meaning foods where the fiber, the protein, and most of the nutrients have been removed during processing, glucose enters your bloodstream very rapidly. Think white bread, crackers, sweetened beverages, most packaged snack foods, white rice eaten in large quantities, pastries. The speed of glucose entry matters enormously. When glucose arrives slowly, as it does from whole foods with intact fiber and protein, your pancreas has time to release insulin proportionally. The response is measured. Blood sugar rises gently and returns to baseline smoothly. When glucose arrives rapidly, the pancreas has to respond aggressively, blood sugar spikes, insulin surges. The overcorrection then causes blood sugar to drop, sometimes below where it started, which triggers hunger, cravings, and fatigue. This is the blood sugar roller coaster that so many people describe, and it is not a willpower problem. It is a predictable physiological response to a specific type of food. Now, here's what makes this particularly significant after 40. As insulin sensitivity declines, the body is less tolerant of these sharp glucose spikes. A meal that your body handled without incident at 28 creates a much larger and more prolonged metabolic disruption at 48. The buffer is smaller. The recovery is slower. The consequences are more noticeable. There's also fascinating research on meal sequencing. Studies from Weill Cornell Medical College found that eating vegetables and protein before carbohydrates at the same meal produced 29% lower blood sugar and 37% lower insulin levels compared to eating the carbohydrates first. Same food, same calories, different order. Profoundly different metabolic outcome. This is one of the most practical and immediately actionable findings in recent nutritional science, and it requires no restriction, just sequencing. Another important dimension here is meal timing and the body's circadian insulin sensitivity. Your cells are significantly more sensitive to insulin in the morning and less sensitive in the evening. This is biological, not behavioral. Your metabolism is calibrated to the light-dark cycle, and insulin sensitivity follows that pattern. Studies have consistently found that eating the same meal in the morning produces a lower blood sugar response than eating that same meal at night. People who eat the majority of their carbohydrates late in the evening are working against their own biology. Not because evening eating is inherently wrong, but because the metabolic machinery is operating in its least efficient mode. The fifth major factor is gut health and its metabolic consequences. The gut microbiome is one of the most rapidly evolving areas of metabolic research, and what scientists have found in the past decade has genuinely changed how we understand insulin resistance. Your digestive tract is home to approximately 38 trillion bacteria. That is a number so large it is almost impossible to conceptualize. And these bacteria are not passengers. They are active participants in your metabolism. They produce compounds that enter your bloodstream. They regulate immune function. They produce neurotransmitters. They influence hormone levels. And they have a direct and documented relationship with insulin sensitivity. Studies comparing the gut bacteria of people with insulin resistance to people with healthy metabolic function consistently find significant differences. People with insulin resistance tend to have lower diversity overall, meaning fewer different species. They tend to have less of certain beneficial strains, particularly bacteria from the Akkermansia and Bifidobacterium families that have been specifically linked to gut barrier integrity and metabolic health. And they tend to have more of bacterial strains that produce inflammatory compounds. One mechanism that has received enormous research attention is what happens when the gut barrier is compromised. The gut lining is a single cell layer thick in most places. When it is healthy, it selectively allows nutrients to pass through while keeping bacteria and their byproducts contained. When it becomes compromised, a condition sometimes called increased intestinal permeability, bacterial fragments called lipopolysaccharides can pass into the bloodstream. Lipopolysaccharides are powerful activators of the immune system. When they enter the blood, the immune system treats them as an infection threat and mounts an inflammatory response. This drives exactly the chronic low-grade inflammation we discussed earlier that directly blocks insulin signaling. Research has found that people with elevated blood levels of these bacterial fragments have significantly higher rates of insulin resistance and metabolic syndrome. And here is what is striking. This can occur even when someone appears to be eating reasonably well. If the gut barrier is compromised and bacteria are leaking through, the inflammatory signal is continuous regardless of diet quality. What damages the gut barrier? Chronic stress is one of the most powerful drivers. It directly alters the tight junctions between gut cells. Poor sleep is another. Antibiotic use, which most adults have had repeated courses of over a lifetime. Diets low in fermentable fiber, which the beneficial bacteria depend on as their primary food source. High alcohol intake, certain common pain medications taken regularly, and low intake of specific nutrients that maintain the structural integrity of the gut lining, including one in particular that connects every single one of these 5 factors together. I have been building to this. And now I want to take the time to explain it properly because this is the part where everything starts to connect. Magnesium. I want to be precise about what I mean when I say magnesium is foundational to everything we have discussed. I am not using hyperbole. The evidence is specific, well documented, and spans decades of research across multiple areas of physiology. Magnesium is involved in more than 300 enzymatic reactions in the human body. An enzyme is a protein that facilitates a chemical reaction. When we say magnesium is a cofactor for over 300 enzymes, we mean that over 300 separate biological processes cannot operate properly without adequate magnesium present. This includes DNA synthesis, protein production, nerve signaling, muscle contraction, heart rhythm regulation, and critically, energy metabolism and insulin signal processing. Let me walk through exactly how magnesium connects to each of the 5 factors we discussed. Starting with insulin signaling itself. When insulin attaches to its receptor on the cell surface, the receptor undergoes a change that activates an internal sequence of reactions. This sequence is called the insulin receptor tyrosine kinase cascade. It sounds complicated, but the key thing to understand is that several of the enzymes in this cascade are magnesium dependent. Without adequate magnesium, these enzymes function below capacity. The signal gets processed inefficiently. The door opens slower. Less glucose is absorbed, and insulin resistance is the result. This is not a peripheral connection. Magnesium is structurally required for insulin's signal to be processed properly inside the cell. Multiple human studies have confirmed this. A comprehensive review published in the World Journal of Diabetes analyzed data from numerous clinical trials and found consistent evidence that magnesium deficiency impairs insulin receptor function and that correcting the deficiency improves insulin sensitivity in a measurable and statistically significant way. The 71% figure I mentioned at the beginning of this video comes from a study that followed people with metabolic syndrome for 1 year. The group with the highest magnesium intake had a 71% lower risk of worsening insulin resistance compared to the group with the lowest intake. A separate 20-year longitudinal study following over 4,000 adults found that higher magnesium intake was consistently and independently associated with lower rates of developing type 2 diabetes. These are different study designs, different populations, different time periods, all pointing in the same direction. Now let us look at magnesium and muscle. Your muscle cells are among the most energy-demanding cells in the body, and they are among the most magnesium dependent. Magnesium is required for a molecule called ATP, which is the actual energy currency that every cell uses to do work. ATP must be bound to magnesium to be biologically active. In other words, without magnesium, your cells cannot even use the energy they produce. They generate it, but they cannot access it. Magnesium is also required for muscle protein synthesis, which is the process by which muscle tissue is built and repaired after exercise. It plays a direct role in how muscles contract and relax, and deficiency contributes to muscle cramps, weakness, and slower recovery after physical activity. This means that if you are making the effort to do resistance training in order to preserve muscle and improve insulin sensitivity, but you are magnesium deficient, the downstream benefits of that effort are partially blocked. You are building with suboptimal materials. The muscle repair is less efficient. The metabolic signaling from muscle contraction is weaker. You work hard and get less return from it. Now, magnesium and sleep. This is the connection that surprises most people and that I find among the most clinically significant. Magnesium plays a direct role in activating the parasympathetic nervous system, which is your rest and recovery mode. It activates receptors called GABA receptors, which produce a calming, settling effect on the nervous system. GABA is the primary inhibitory neurotransmitter in the brain. When it is active, your mind quiets, overstimulation decreases. The body begins preparing for rest. Magnesium also helps regulate melatonin production. Melatonin is the hormone your brain produces in response to darkness that signals the body to shift into sleep mode. The enzyme that converts the precursor molecule into melatonin requires magnesium as a cofactor. Low magnesium can impair this conversion, leading to lower melatonin levels and difficulty initiating sleep. And magnesium directly modulates cortisol. It inhibits the release of cortisol from the adrenal glands and dampens the sensitivity of cortisol receptors. This is particularly significant at night when cortisol should be at its lowest. In magnesium-deficient individuals, cortisol tends to stay elevated longer into the evening, making it harder to fall asleep and maintaining the metabolic disruption that high cortisol creates. Multiple clinical trials have found that magnesium supplementation improves sleep onset, sleep duration, and sleep quality, with particularly strong effects in adults over 50. One randomized controlled trial published in the Journal of Research in Medical Sciences found that older adults who supplemented with magnesium fell asleep faster, slept longer, woke up less frequently during the night, and had lower cortisol levels in the morning compared to the placebo group. Now consider the chain. Magnesium deficiency impairs sleep. Poor sleep worsens insulin resistance. Insulin resistance disrupts cortisol regulation. Elevated cortisol further depletes magnesium by increasing urinary excretion. Lower magnesium impairs sleep further. The cycle continues, deepening with each rotation. This is why addressing magnesium can sometimes be the single intervention that begins unraveling the entire cycle. Not because it fixes everything, but because it removes a core constraint that was blocking improvement across multiple systems simultaneously. Now, magnesium and inflammation. There is a well-established inverse relationship between magnesium levels and markers of systemic inflammation. Studies consistently show that low magnesium is associated with elevated levels of C-reactive protein, which is the most commonly used blood marker for chronic inflammation, as well as elevated TNF-alpha and interleukin-6, the specific cytokines that block insulin receptor function. The mechanism is related to magnesium's role in regulating NF-κB, which is a master switch that controls inflammatory gene expression. When magnesium is adequate, NF-κB is kept in check. When magnesium is low, NF-κB becomes more active, and the inflammatory signaling cascade it controls amplifies. This leads to chronically elevated cytokines, impaired insulin signaling, and faster accumulation of visceral fat. A meta-analysis published in Nutrition Reviews analyzed data from multiple studies and found that higher magnesium intake was independently associated with lower concentrations of inflammatory markers. This held true across different populations and different methods of measuring inflammation. The signal was consistent and meaningful. Now, magnesium and the gut. Magnesium has several direct effects on gut health that most people have never heard. The cells that line the gut wall, called enterocytes, depend on magnesium for their structural integrity and their energy production. The tight junctions between gut cells that prevent bacteria from leaking through are partly regulated by magnesium-dependent enzymatic processes. When magnesium is low, the gut barrier becomes more permeable, more prone to allowing bacterial fragments through into the bloodstream, and more prone to chronic inflammation. Magnesium also regulates gut motility, meaning how efficiently food and waste move through the digestive tract. Slow motility allows more time for fermentation of food by gut bacteria, including potentially harmful strains, and this can shift the microbial balance over time. And the beneficial bacteria in your gut, the strains that produce anti-inflammatory short-chain fatty acids and support insulin sensitivity, are more abundant in environments with adequate mineral availability. Magnesium is among the minerals they depend on, so the picture is complete. Magnesium deficiency quietly degrades insulin signaling. It impairs muscle recovery and function. It disrupts sleep and elevates cortisol. It amplifies chronic inflammation. And it compromises gut barrier integrity. All 5 of the factors that drive insulin resistance after 40 are directly connected to the adequacy of this single mineral. And the data on how common magnesium deficiency is in modern populations is alarming. National nutrition surveys in the United States and across Europe consistently find that between 50 and 60% of adults consume less magnesium than the recommended daily intake. And the RDA itself, the recommended daily allowance, is likely set conservatively for people who are metabolically healthy. For someone who is under chronic stress, exercising regularly, consuming a diet with significant processed food content, or dealing with any degree of insulin resistance, actual magnesium requirements are higher. Here is why the deficiency is so pervasive. Modern agricultural practices have depleted magnesium from topsoil over the past several decades. Vegetables grown in magnesium-depleted soil contain less magnesium than the same vegetables grown 50 years ago. Studies comparing mineral content in produce over time show reductions of 20 to 30% for some vegetables. Processed and refined foods, which constitute a significant portion of many modern diets, are largely stripped of magnesium during manufacturing. White flour, for example, retains only about 15% of the magnesium present in whole grain wheat. Refining removes the bran and germ, which are the primary magnesium-containing portions of the grain. Certain very common factors actively deplete magnesium from the body. Chronic psychological stress increases urinary magnesium excretion. Studies have shown that physical or emotional stress can double the rate at which magnesium is lost through urine. High sugar intake, both from foods and from elevated blood sugar and insulin resistance, also increases urinary magnesium loss. Heavy caffeine consumption has a mild diuretic effect that contributes to magnesium loss. Alcohol significantly increases renal excretion of magnesium. And certain commonly used medications, including proton pump inhibitors for acid reflux and some diuretics and blood pressure medications, can substantially lower magnesium levels. The result is that many people are losing magnesium faster than they are replacing it, and doing so while simultaneously having elevated requirements due to metabolic stress. And because standard blood tests measure serum magnesium, meaning the magnesium circulating in the blood, rather than intracellular magnesium, which is where 99% of the body's magnesium is stored, deficiency often goes completely undetected on routine labs. You can be functionally deficient in magnesium and have a serum magnesium level that appears normal. This is why I want you to understand the concept of subclinical deficiency. A deficiency does not have to be severe enough to cause dramatic symptoms to impair biological function. Even operating at 70 or 80% of optimal magnesium status creates measurable impairment in insulin signaling, sleep quality, inflammatory regulation, and muscle recovery. You will not feel dramatically ill, but you will feel like everything is slightly harder than it should be, like your body is not quite responding the way it used to. That is the experience. And for millions of people, this is a significant contributing factor to the metabolic decline they are experiencing. Now, let me be very clear about something important. I am not telling you that magnesium is a cure for insulin resistance. Nothing I have described today works in isolation. The body is a system, and systems require comprehensive support. What I am telling you is that for many people, magnesium deficiency is a key missing piece that makes every other effort less effective than it should be. Addressing it does not replace the need for movement, sleep, stress management, and dietary improvement, but it creates better biological conditions for all of those things to work. Let me now talk about what dietary sources of magnesium actually look like in practice, because this is where the information needs to become actionable. The most magnesium-dense foods are dark leafy greens, particularly spinach, Swiss chard, and kale. A single cup of cooked spinach provides about 157 milligrams of magnesium. Pumpkin seeds are extraordinarily dense, providing about 150 milligrams per ounce, making them one of the most concentrated dietary sources available. Black beans and lentils provide 60 to 70 milligrams per half cup. Almonds and cashews provide 60 to 80 milligrams per ounce. Avocado provides about 58 milligrams in a medium-sized fruit. Dark chocolate with 70% or higher cocoa content provides about 64 milligrams per ounce and has the added benefit of flavonoids with anti-inflammatory properties. The adult recommended daily intake is approximately 420 milligrams for men and 320 milligrams for women. But as I mentioned, under conditions of metabolic stress, actual optimal intake is likely higher. For many people, closing the gap through diet alone requires a consistent and intentional effort to include these foods daily. That is absolutely worth pursuing. But for people who are under significant metabolic stress, who have been deficient for an extended period, or who have lifestyle factors that increase magnesium excretion, supplementation is often a practical and evidence-supported step. If you are considering supplementation, the form matters considerably. Magnesium oxide is the cheapest and most common form found in general multivitamins, but it has poor absorption. Roughly 4% is absorbed in the gut. Magnesium citrate is better absorbed and is often used for general supplementation. Magnesium glycinate, where magnesium is bound to the amino acid glycine, is highly bioavailable, well tolerated by the digestive system, and has the added benefit of glycine itself, promoting relaxation and sleep quality. Magnesium malate, bound to malic acid, is often used specifically for energy and muscle function because malate is involved in the energy production cycle inside mitochondria. For people with insulin resistance and fatigue, magnesium malate or a combination of glycinate and malate is often a logical choice. Standard supplemental doses in research range from 200 to 400 milligrams per day, typically taken in the evening. If you are considering supplementation, discussing the appropriate form and dose with your healthcare provider is a reasonable step, particularly if you have kidney function concerns, as the kidneys regulate magnesium excretion and any kidney impairment changes the picture. Now, I want to broaden the conversation and talk about several other nutritional factors that are important in the insulin resistance picture and are often missed. The first is chromium. Chromium is a trace mineral that plays a specific role in carbohydrate metabolism. It is a component of a molecule called chromogulin, which amplifies the activity of insulin receptors. When chromium is adequate, insulin receptors respond more sensitively and efficiently to insulin signals. Research has found that chromium supplementation in people with insulin resistance and type 2 diabetes can meaningfully improve fasting blood glucose and insulin sensitivity. Dietary sources of chromium include broccoli, which is one of the richest sources, whole grains, green beans, beef, and turkey. Chromium, like magnesium, is depleted by high sugar diets and by chronic stress, and standard blood tests do not reliably measure chromium status. The second is vitamin D. Vitamin D is not just a bone mineral, it functions more like a hormone than a vitamin, and its receptors are found in virtually every tissue in the body, including the pancreatic cells that produce insulin. Studies have consistently found that vitamin D deficiency is associated with higher rates of insulin resistance and type 2 diabetes. Research suggests that vitamin D plays a role in reducing inflammation, in supporting beta cell function in the pancreas, and in directly improving cellular insulin sensitivity. Vitamin D deficiency is extraordinarily common, particularly in northern latitudes, in people who spend most of their time indoors, in people with darker skin tones, and in older adults whose skin becomes less efficient at producing vitamin D from sunlight. Many clinicians consider optimal vitamin D status, blood levels above 40 nanograms per milliliter, to be a foundation of metabolic health support. The third is omega-3 fatty acids. The long-chain omega-3 fats found in fatty fish, EPA, and DHA have robust evidence for anti-inflammatory effects. They work partly by incorporating into cell membranes and changing how receptors on the cell surface function. Cells with higher omega-3 content in their membranes are more fluid and more responsive to insulin signals. Omega-3 fats also reduce the production of inflammatory cytokines, directly addressing one of the core drivers of insulin resistance we discussed. A diet with regularly consumed fatty fish such as salmon, sardines, mackerel, and herring provides meaningful omega-3 support. For people who do not eat fish regularly, high-quality fish oil supplementation is well supported in the research literature. Now, let me spend time on the practical protocol because everything I have described needs a real-world structure. The morning matters enormously for metabolic health. When you wake up, your cortisol is naturally at its highest point of the day. This is normal and appropriate. But what you do in the first 2 hours of your morning either supports or disrupts the hormonal environment for the entire day. Morning light exposure is one of the most underused metabolic tools available. Getting natural light into your eyes within the first hour of waking, even on a cloudy day, sets your circadian clock, begins the cortisol clearance process, and synchronizes the timing signals that govern insulin sensitivity throughout the day. This is not about staring at the sun. It is about getting outside or near a window without sunglasses for even 10 to 15 minutes. Morning movement is the other key. Even a 10-minute walk after breakfast produces a measurable reduction in post-meal blood sugar compared to sitting. The muscle contractions activate glucose transporters in muscle cells independent of insulin, allowing glucose to enter cells through an alternate pathway. For people with insulin resistance, this is powerful. You're using the glucose before it has time to drive an insulin surge. Breakfast composition matters. Starting the day with protein first, even 20 to 30 grams, stabilizes blood sugar for the entire morning. It reduces the midday craving cascade. It supports muscle protein synthesis during the period when the body's anabolic signals are naturally strongest. Eggs, Greek yogurt, cottage cheese, a protein smoothie with minimal added sugar, these are practical high-protein morning options. Afternoon is when most people hit their metabolic wall. The 2 to 3 p.m. energy crash is not imaginary, and it is not just from lunch. It is partly circadian. There's a natural dip in alertness in the early afternoon, but it is significantly amplified by blood sugar instability from poorly structured morning and midday eating. If you find yourself reaching for caffeine or sugar at 2 p.m. routinely, the issue is almost certainly upstream, in what you ate in the morning and at lunch, not in the afternoon itself. A 5 to 10-minute walk after lunch has been shown in research to significantly reduce post-meal blood sugar and improve afternoon energy. It requires no equipment, no gym, no program, just movement shortly after eating, which is when the muscle's glucose disposal capacity is most useful. Resistance training, ideally on 2 to 3 non-consecutive days per week, is the cornerstone of the movement strategy. You do not need a full gym. A set of resistance bands and knowledge of basic compound movements, squats, lunges, rows, presses, hip hinges, is sufficient to create the hormonal and metabolic stimulus your body needs. Compound movements that involve multiple large muscle groups create the largest metabolic and hormonal response. Protein intake throughout the day, not just at one meal, supports muscle protein synthesis more effectively than concentrating protein in a single meal. Research on muscle protein synthesis shows that the body can effectively use approximately 30 to 40 grams of protein per meal for muscle building purposes. Larger amounts at one sitting do not proportionally increase the benefit. Spreading protein across 3 meals optimizes the signal. Evening is where metabolic discipline becomes particularly important. The body's insulin sensitivity is at its daily low in the evening. Heavy carbohydrate meals late at night create a metabolic burden that the body is not optimally equipped to handle. This does not mean no carbohydrates at dinner. It means smaller quantities, paired with protein and vegetables, and ideally eaten before 7 p.m. if possible given your schedule. Artificial light in the evening suppresses melatonin and delays the circadian signals that prepare the body for sleep. Screens, overhead lighting, and particularly blue light wavelengths from devices tell the brain it is still daytime. Using blue light filtering settings on devices after 8 p.m., dimming household lighting, and establishing a consistent wind-down routine are not just comfort habits. They are biological inputs that directly affect cortisol regulation, melatonin production, and ultimately the quality of sleep that governs insulin sensitivity the next day. And this brings me to what I consider one of the most important concepts in metabolic health: the compounding effect of daily habits. Every single thing I have described today, the morning light, the post-meal walk, the resistance training, the protein distribution, the sleep hygiene, the evening light reduction, and the nutritional support, including adequate magnesium, none of them produce dramatic results in isolation or in a single day. But when they are practiced consistently, they compound. Each one shifts the biological environment slightly, and the biological environment shifts compound on each other. Week 1, sleep quality begins to improve. Cortisol starts to regulate better. Week 2, post-meal energy crashes begin to reduce. Cravings are slightly quieter. Week 3, morning energy is more consistent. The afternoon wall is less severe. Week 4 and 5, the body composition begins to shift. Not dramatically, but the direction has changed. By month 3, the person looks back and cannot identify the single moment things turned around, because there was not one. There were hundreds of small moments, each adding to the previous, and the cumulative biology is fundamentally different. This is what real metabolic recovery looks like. Not a dramatic transformation in 21 days. A gradual compounding shift in how your body operates at the cellular level. And the fact that it is gradual does not make it less powerful. It makes it more durable. I want to address one more thing that I think deserves direct acknowledgement. For many people, the hardest part of this journey is not finding the information. It is maintaining belief that the effort will produce results when the results are not immediately visible, and that difficulty is legitimate. It is not weakness. It is a natural response to having tried things before that did not work. The difference now, I hope, is understanding. When you understand that your cells need specific nutritional support to even respond properly to insulin signals, you stop wondering why calorie restriction alone did not work. When you understand that sleep deprivation measurably drops insulin sensitivity by 25%, you stop wondering why the diet that worked in your 30s is not working now. When you understand that chronic inflammation is a mechanical barrier to insulin receptor function, you stop assuming that trying harder will somehow override the biology. You are not fighting your body. Not. You are learning to speak its language, and the body is remarkably willing to respond when you do. The symptoms that feel like permanent damage, the belly fat, the fatigue, the brain fog, the stubborn weight, they are not your destiny. They are a description of your current biological state. And biological states can change. I want to leave you with a clear, practical summary that you can actually use. First, protect and build your muscle. 2 to 3 sessions of resistance training per week is not optional for metabolic health after 40. Muscle is the primary site of glucose disposal and the primary source of metabolic signaling. Use it. Second, treat sleep as medicine. 7 to 8 hours in a dark, cool room with consistent timing is one of the most powerful metabolic interventions available. Blue light management in the evening and morning light exposure both support circadian biology that directly governs insulin sensitivity. Third, restructure your eating patterns. Structure your eating, not restrict. Restructure. Eat vegetables and protein before starches. Prioritize earlier meals over later ones. Reduce quickly absorbed refined carbohydrates and increase fiber, protein, and healthy fat to slow glucose absorption. Walk for 5 to 10 minutes after meals when possible. Fourth, address the inflammatory environment. This means reducing the inputs that drive chronic inflammation and increasing the inputs that resolve it. Movement, sleep, a diet rich in vegetables and anti-inflammatory fats, and stress management all contribute. Fifth, check your mineral and nutrient status. Magnesium, vitamin D, chromium, and omega-3 fatty acids are 4 of the most well-evidenced nutritional factors in insulin sensitivity. Dietary sources matter. For people under metabolic stress, targeted supplementation is often a reasonable support strategy. And sixth, be patient with the timeline and ruthless with the consistency. Metabolic recovery is not a 21-day project. It is a 6-month, 12-month ongoing commitment to giving your body what it needs to function properly. The compound interest of consistent daily action is more powerful than any dramatic short-term intervention. You are not too far gone. You are not too old. You are not permanently broken. You are a biological system that has been under compounding stress without adequate support. And systems, when given the right conditions, are capable of recovery that genuinely surprises people. That is what I have seen over 20 years of working with patients who came to me convinced they had run out of options. And some of the most meaningful progress I have ever witnessed came from people who were simply told the truth about what was happening in their biology and given a framework they could actually use. That is what this video has been. A framework grounded in the actual science of how your metabolism works and what it needs to work better. If you want to go deeper and get a detailed step-by-step guide built around everything we discussed today, I have linked a comprehensive longevity resource in the description below. It is free, and it goes further than what we have been able to cover here. And if any part of today's video connected with something you have been experiencing, I genuinely want to hear about it in the comments. What was the thing that finally made sense? What have you been struggling with that we addressed today? Your story matters. And it may be exactly what another person watching this needs to read. Thank you for giving me your time today. I know you could have spent it anywhere. Take care of your body. It is more capable of recovery than you think. And I will see you in the next video.